In this video, we will cover insight about the changes within the immune response and the cancer cells which change in the stiffness of the ECM. Although we have covered this topic in our previous videos, in this video we will go in deep and will understand which process of the immune response get affected and how it will trigger the intercellular changes, or what are the intercellular changes occurs because of the immune response, because of that immune response, and all these things are being related to the stiffness of the ECM. So, and this is the brief overview that we have covered in our previous videos. So, in this case, the ECM is stiff. The stiff ECM will interact with the integrins on the cells. It will interact with the integrins on the cells, and that will trigger this ERK phosphorylation of this ERK pathway that will cause this effect in polymerizations, and that will lead to this ammovia like moving, and that will cause this infiltration and migrations of the cells. So these things we have covered, and in this case occurs because when the ECM is stiff. So now let us come back to our main topic of this video. Uh, this this cancer cells, suppose these cancer cells are present within the host, and if the ECM is soft, then the immature dendritic it will increase, or the movement of the immune cells will be high in the soft ECM. In that case, the immature dendritic cells will recognize these cancer cells and provide this antigen informations about the cancer cells to the T cells. In this process, the immature dendritic cells will become the mature dendritic cells. And also, since the ECM is soft, the migration of these mature dendritic cells will be dendritic cells will be easier, and it will increase the efficiency of the immune response. And once this information has been provided to the T cells, and right now the T cells is in naive stage. So in order to move from T cells from naive stage to the effector cells, if the stiffness is stiffness of the ECM is high, then this step will get been will be efficiency of this step will be reduced. And if the stiffness of this ECM is low, then the conversion from naive T cells to the T effector cells will be high. So this is one of the steps where immune response get hindered. So after this T effector cells, it can move. It can one way is that it can move to the CD8 plus T cells that has the informations or that has the informations about the this unique epitopes that are expressed on the cancer cell surface. And if it is a soft ECM, then this CD8 plus T cells activations will be easier, and the cytotoxicity killing will be higher in case this ECM is the soft. However, when the ECM is stiff, then it will leads to then if the ECM is stiff, then it will the cytotoxicity of this CD8 plus T cells will be reduced, as the movement of these T cells in the stiffer ECM will be lower. At the same time, what will happen is that the stiffer ECM, or the cytokines that are released by cancer cells, will lead to the formation of this cancer associated fibroblast, and this cancer associated fibroblast will neutralize this neutrophil in terms of its survival activations, chemotaxis, and extracellular traps will be reduced. That neutrophils takes part to take care of the harmful epitopes that have been detected by the immune system of the host. And along with that, what will happen is that this cancer associated fibroblast will also inhibit this CDT cells antigen processing, so that its activation will get reduced. And if when the CD8 the T effector cells will convert it into this CD4 plus T cells, then what will happen? One pathway for the T cells effector is to go towards CD8 plus T cells. Another pathway is that it will go either towards TH1 or TH2 formations. So this naive CDT plus cells, when these cancer associated fibroblasts are produced, that it will promote the migration of this naive CD4 plus T cells. And in case of this soft ECM, what it will does is that it will leads to this TH1 formations, and that will cause this M1 macrophage formations, and it will has this anti-tumorigenic effect, such as that it will trigger this phagocytosis within the cancer cells. However, the conditions where this cancer-associated fibroblast form and the soft ECM is uh, close to hypothetical conditions, as the tumor microenvironment around the cancer cells has this cancer-associated fibroblast and the stiff ECM. So when this stiff ECM conditions are there, then it will trigger this naive CD2 plus cells to migrate towards this stiff ECM phase, to migrate towards this stiff ECM phase. So one thing that we need to understand is that this conversion from M0 phase to M2 phase occurs of this M0 macrophage, and this Th2 and Th1 accumulate or act as a catalyst to help this macrophage to convert it into the next differentiate into the next macrophage. So in case of this soft ECM, the Th1 helps this M0 macrophage to differentiate to the M1 macrophage, which has the anti-tuberogenic properties. And in case of this stiff ECM, the M0 macrophage will differentiate this into the M2 macrophage with the help of this Th2 cells. So this M2 macrophage will have this anti-tumorigenic properties, and it will promote the pro-inflammatory. It will promote not pro-inflammatory. It will promote the pro-tumorigenic response, or more survival genes, and at the same time there is more genes that will promote the migration or metastasis of the tumor cells. And how this NK cells plays a role. This NK cells functions will be reduced when by this cancer associated fibroblast, as it will reduce the cytotoxicity or cytokine productions by the NK cells, and thereby also this NK cells functions against the tumor cells will be reduced. And overall this M2 macrophage has this pro-tumorigenic properties, immunosuppressions, wound healing, and anti-inflammatory properties, whereas anti-tumorigenic M1 macrophages has this anti-tumoral immunostimulation, tissue injury, or pro-inflammatory properties. And these are the cytokines that are released by M1 macrophage, and these are the cytokines that are released by M2 macrophages. So I hope you right now get some basic insights about what are the processes that get affected by the immune response, and how it will leads to the different intercellular signaling pathways trigger within the cancer cells. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.